Today I'll tell you about clinical biochemistry of kidneys. Plan of the lecture. I will tell you about structure and function of kidneys, urine formation, renal function assessment, and biomarkers of acute kidney injury, biochemical analysis of urine. So these are all topics we will discuss today. Structure and function. Yeah? You know, you already learned structure and function of kidneys on physiology, anatomy, histology, right? So we will repeat right now some key points. Look, functions, the main functions are excretory, so excretion of components which we don't need, like urea, uric acid, excessive ions, and water from the body. The second function is a regulatory function, regulation of pH and ionic composition of blood. So kidneys can excrete hydrogen ions. Regulation of water balance. That means that kidney can excrete an excess of water. And the regulation of red blood cells production. That means that it can produce signal molecules, erythropoietin, which induces production of new red blood cells. Okay. The last function is detoxification. It's a neutralization of endogenous and exogenous antibiotics and synthetic compounds. That means that if we get some toxic compound, we have to excrete it, and at the first stage of excretion is oxidation and conjugation, and excretion, the last stage. So kidneys, structure of kidneys. Here you can see two kidneys here at the left picture, and kidney has a good position because it interacts with water and get approximately 2 times more oxygen compared with brain, because the main function of kidneys is a transport of ions, and that process need to more energy if any other types of process. From kidney, here you can see ureter, bladder, and ureter. This is excretory system. On the right picture, you can see renal cortex here, renal blood vessel here, renal medulla here, renal pelvis. This one pelvis is a free space for accumulation of urine. Renal artery here, ureter here. So and this is the macroscopic structure. Here you can see the structure of nephron. It contains several key parts. The first part of nephron is glomerulus here, glomerulus. And after the glomerulus, you can see proximal convoluted tubule here, loop of Henle here, and here distal convoluted tubule. So why this structure is very important, and because to understand how kidneys produce, nephrons produce urine, we have to know what is the main function of different components of nephron here and here. So to understand this, we have know that glomerulus get blood from artery here, red, colored by red, and it can into glomerulus enters afferent arteriole here, and afferent arteriole here. After exit from glomerulus, afferent arteriole go and surrounds the structure of tubule and works like a reservoir for different components of primary urine. And these components like glucose, amino acids, transported from urine into bloodstream. Functional parts of the nephron. Glomerulus. The main function of the glomerulus is blood filtration. So difference between diameters between afferent and efferent arterioles works like a structure which can form a blood filtration does blood filtration. Tubules. The main function of tubules is reabsorption of components of very useful components into blood. That means that after filtration, some small molecules like glucose, some useful small molecules, glucose, maybe some ions, like iron, like amino acids, we have, because they were filtered into tubules, but they are very useful, and our kidneys have to reabsorb it into blood. And the last component of nephron is a collecting tubule here. The main function of collecting tubule is water reabsorption. That means that we can produce approximately 200 liters of primary urine, but excrete outside only approximately 2 liters secondary urine, which contains only waste products. But useful products should be reabsorbed. Urine formation. Here you can see afferent and efferent arteriole, bone space, proximal tubule. And the main way of production of primary urine is the filtration, glomerular filtration here. And because afferent and efferent arterioles have different diameters, so excess of water which contains solid ions, small organic molecules water transferred into this space, bone man's space here, into this space here. This process, glomerular filtration, is a passive process. So we don't have to spend ATP to do this work, because only different diameters of these arterioles can works like a force which transport components of blood into Bowman's space. Result of primary information is formation of approximately 200 liters per day of primary urine. Juxtaglomerular apparatus here. This one. The main idea of regulation, formation of urine, our kidneys have to, our nephrons have to know, do they have to increase filtration pressure or not? So how does it work? Here you can see a distal tubule here. This one glomerulus, and this one, this small part, triangle. See several cells. This is macula densa. It's Latin words. Macula means spot. Densa means density. So density, and the cells works like sensors for estimation of rate of filtration. And if a rate of filtration is very low, the cells are should be activated and produce some prostaglandins, which activate production of renin inside this violet cells. This one. And after that, renin enters into the afferent arteriole and works like an enzyme which activate conversion of angiotensinogen into angiotensin 1, and after conversion of this molecule into angiotensin 2. The result is a contraction of afferent arteriole, this one. The whole result is an increasing of difference between diameters of afferent arteriole, this one, and afferent, this one. Afferent and afferent, this one. And the result of this, the end result of this action is the production more and more primary urine. Reabsorption, the second stage of working of nephron. So here you can see different colored by different colors sites of nephrons, and you can see in the proximal tubule reabsorbed sodium, chloride, potassium, ions, water, glucose, amino acids, and secreted some amount of hydrogen atoms and organic acids. This site of loop of Henle, this site of reabsorption of water and reabsorption of several types of ions, like sodium, chloride ions, potassium, calcium, and secretion of hydrogen atoms. The last part of nephron is a regulated part. In this space, takes place a process which regulated by reabsorption, which regulated by different types of hormones, like antidiuretic hormone produced by hypothalamus. It's regulated by aldosterone. This site is regulated by sodium uretic hormone here, and the result of reabsorption is a formation of small amount of urine, approximately 2 liters. Renal function assessment and biomarkers of acute kidney injury. The main marker of rate of filtration is a glomerular filtration rate, this one. How to estimate the glomerular filtration rate? We use chemical creatinine, which we can estimate the rate of filtration of creatinine from blood into urine. How can we do this work? Because the rate of filtration of creatinine is dependent on only the rate of filtration. This chemical does not reabsorb it, does not secrete it, and this creatinine only filtrated. And concentration of creatinine in primary urine is equal to the concentration of creatinine in the blood plasma. So if we know the volume of urine and concentration of creatinine in urine and concentration of creatinine in blood, we can estimate the rate of clearance of creatinine. Here, glomerular filtration rate, here, milliliters per minute, is equal concentration of ureate creatinine times diuresis divided by concentration of plasma creatinine. So we can estimate the general function of kidneys filtration. In case of lowering of glomerular filtration rate, these patients suffer from acute kidney injury. So this one, we use creatinine concentration as in blood, as a marker of function of kidneys. So increase in serum creatinine times to 1.5 means a risk factor or first stage of kidney failure. The second stage or injury, increase in serum creatinine 2 times, more than 2 times. The third stage of kidney damage is increase in serum creatinine times 3. And loss of function, complete loss of kidney function more for weeks, and end stage kidney disease more than 3 months. Here, we can use not only creatinine concentration, but we can estimate a volume of urine, secondary urine. And here at the left part of this table, you can see different stages of kidney failure. Here, we know several types of molecules which we can use to estimate kidney injury. The first one is a kidney injury molecule 1, or KIM. This molecule is a membrane glycoprotein, and it expresses by epithelium of kidney. And after destruction, after damage of kidney, this molecule transported into urine, and if we estimate concentration of KIM1 in urine, we can estimate the stage of, or the fact of damage of kidneys. The second molecule is a neutrophil gelatinase-associated lipocalin. This one is a small protein which synthesizes and induces it in the epithelium of the kidney tubules. In case of damage of kidneys, the concentration of NGAL, this molecule, increases in urine. Why this molecule better than creatinine? Because concentration of all these molecules, like KIM1 and this one, increase it before changing of concentration of creatinine, and we can give this patient treatment before, if even he doesn't feel anything bad. The third molecule, interleukin 18. This molecule, interleukin, induces of gamma interferon synthesis. That means that it works like a molecule which activates antiviruses immune reaction. And in case of kidney, this molecule induced after ischemia and reperfusion. Yeah. And again, the concentration of this interleukin in urine, we can find before from 1 to 2 days before changing concentration of creatinine in blood. So it's much better because in this case we can give this patient specific treatment. The last molecule is fatty acid binding protein. This protein, the main function of this protein is to transport lipophilic or hydrophobic molecules inside cells, because cells contains membranes divided by water, and some hydrophobic molecules could not be transported from one side of cells into another side of cell. And to do this work, our cells, most of our cells, express these proteins, fatty acid binding protein. The name fatty acid means that this protein was found as a transporter for fatty acids, but it can transport any hydrophobic molecule, even small molecule, large molecule. Increasing of the concentration of fatty acid binding protein in the urine is a marker of acute kidney injury. Again, from 1 to 2 days before the increase in the concentration of creatinine in the blood. So it's a useful marker for diagnostic of acute kidney injury. Biochemical analysis of urine. So we will discuss some cases in class, and we have to know some components which we use to provide true diagnostics. Proteinuria, this one, is a situation of accumulation of protein in urine. We know 3 types of proteinuria: pre-renal, so before kidney; renal, kidney; and post-renal, after kidney. Pre-renal proteinuria, this one. Paraproteinemic hemoblastosis, like multiple myeloma. That means that this is a situation when patients suffer from tumor which produces inappropriate antibodies. The second situation, the hemoglobinuria, is a situation in which we can find hemoglobin in urine in case of hemolysis, so damage of red blood cells. The last situation is a myoglobinuria. That means that if patients suffer from damage or inflammation in muscles, components of muscles like myoglobin could be transported into blood at first, and after that into urine, filtrated into urine. So this is the pre-renal, so before renal, before kidneys, proteinuria. Renal proteinuria. So problem, it takes place inside kidneys. Glomerulonephritis, this one. Glomerulonephritis. This one is a situation of inflammation inside glomerulus. So the result is broken down of filter and and large proteins could be transported, could be filtered from serum into urine, and we can find the protein in urine. The next one is diabetic nephropathy. This one is a situation when, in case of doesn't depend on type of diabetes mellitus, glucose is a toxic chemical, can damage glomerulus and damage kidneys. As a result, is a formation of filters which can transfer more larger molecules, like albumin. Amyloidosis, this one, is a situation when the kidneys were damaged by some toxic chemicals. And as a result is accumulation of proteins inside epithelial cells of kidneys. And they damage it, and components of the cells transported into urine, and we can find this situation. And toxic nephropathy is an acute damage of kidneys. Post-renal proteinuria. That means that problem of accumulation of protein takes place after kidneys. So the first one, pielitis, this one, cystitis, bladder cancer, ureteritis. That means that after kidneys, maybe it takes place an inflammation and damage by bacterial inflammation and infectious, in case of infectious. And the result is damage of cells, epithelial cells, and transport proteins into urine. We can find post-renal proteinuria. Next marker is the next result is glycosuria. That means that we can find increased concentration of glucose in urine. In healthy people, we can find very small amount of glucose. So we can even say that normal urine from healthy people doesn't contain glucose. But in case of increased concentration of glucose in blood, like in case of diabetes mellitus acute pancreatitis, pancreonecrosis, diabetes mellitus is a deficit of insulin, and as a result is increase in concentration of glucose in blood. Acute pancreatitis is damage of beta cells of pancreatic gland. As a result is accumulation of glucose in blood. And pancreonecrosis, that means that damage of pancreas. As a result is accumulation of glucose in blood, and after filtration, this glucose could not be transported into reabsorbed, could not be transported into blood, and as a result is a glycosuria. That means that we can find high concentration of glucose in blood. The second type of glycosuria, the second reasons of the glycosuria, extrapancreatic glycosuria. That means that some problems which does not relate it with the pancreas, and these are disease of the central nervous system. Some molecules like some hormones can induce production of glucose. Hormonal hyperthyroidism, pheochromocytoma, acromegaly. This situation is these hormones works as activators of production of glucose. Sometimes renal, organic kidney damage. That means that in some cases, damaged epithelium could not be can't transport glucose or even amino acids from urine, from primary urine into blood. Elementary, if you eat 5 kilograms of sugar, the result is glycosuria, because a concentration of glucose in blood become very high, very increased, and the result is a filtration of glucose into your primary urine. And in this case, kidneys could not be transport. They could not transport the glucose back to blood. Some drugs could activate using different mechanisms, can stimulate glycosuria. The next types of components which we can estimate in urine: ketones, bilirubin, urobilinogen. Ketonuria is a situation we can find in case of diabetes mellitus, because of especially type 1 diabetes mellitus, because in this case, in case of deficit of insulin, our brain thinks that it should activate production of energy from in the form of ketone bodies from fatty acids, which are deposited in the form of triacylglycerols in adipose tissue. The next situation is an unbalanced diet or fasting. That means that in case of deficit of food, our brain should activate production of energy. But without glucose, the source of energy, the source of ATP, we also activate production of free fatty acids, and after that ketone bodies. Alcohol poisoning. A situation has two reasons. Because alcohol, after conversion of oxidation, produce acetylcoenzyme A, which works like a substrate for production of ketone bodies. The second reason is a blocking of secretion of insulin, and as a result, our brain thinks that we have no energy and activate lipolysis and stimulate production of ketone bases. Pancreatitis, the similar situation like diabetes mellitus, deficit of insulin, and sometimes infectious diseases which can stimulate ketones, production of ketones, like a part of stress reaction. Bilirubinuria, this one. You know that we have the main two types of bilirubin. The first one is the unconjugated bilirubin, which are produced by spleen, and this molecule should be converted to conjugated bilirubin. And only conjugated bilirubin we can find in urine, because this molecule is hydrophilic. There are two reasons of increasing concentration of bilirubin and conjugated bilirubin and increasing concentration of bilirubin in urine. They are obstructive jaundice. That means that bile could not be transported from liver into small intestine. And the second one, in the parenchymal jaundice. That means that in case of damage of liver cells, some amount of bile could be transported from bile capillaries into blood. The last marker is urobilinogen. That means that some amount of approximately 5% of urobilinogen should be converted into urobilin. And in case of damage of liver, some amount of this component transported into blood and back to the urine. The last components which we can find in urine and make a conclusion: hemoglobin, myoglobin, and nitrites. Hemoglobinuria is a very simple situation. So in case of damage of red blood cells, components of red blood cells transported into serum in free form, and after filtration, hemoglobin could be transported into urine, primary urine. As a result, we can find hemoglobin in urine. The second component, myoglobinuria. That means that in case of damage of muscles, like a result of excessive muscle injuries, myositis, hypertrophy, as a result of the damage of muscle cells, and components from and protein components from muscles could be transported into serum and could be filtered into primary urine. The last component which we can find in urine is a nitrite. The main idea is that bacteria use urea, uric acids, amino acids, could be transferred, they can use them as a source of energy or structural components. And they have to oxidize these chemicals. And if we find high concentration of nitrile in urine, we can conclude that this patient has a bacterial infection. We don't know where this bacterial infection takes place, but we know we have to observe these patients more precisely. Okay? So I think I gave you all information which you need to solve our clinical cases. This information is very useful because you can solve. When we discuss in class, we will discuss some clinical cases which are related with changing concentration of these compounds, different components.